Right, so we are going to talk a lot about DNA today. This is somebody's DNA profile. And what they're actually doing there is typing the DNA, and this would be what would be used to make a match between one DNA sample to another DNA sample. It's called electrophoresis. All right, but anyways, so we're going to be talking a lot about genetics. And genetics, to me, is the best argument that we will have with respect to biological complexity, because there's nothing more complex in the body than your DNA, which we're going to talk about today. So, but before we do that, I want to kind of summarize a little bit. What is the purpose of this section, the creative design section? Even though we took a little detour into intelligent design, I want to bring it back to creative design. Remember, we are creationists, and we're not ashamed of the fact that we believe and are convinced of the fact that God created everything. The other thing that we are not ashamed of is that the book of Genesis is a literal interpretation of the beginning of the universe, the beginning of life, the beginning of humanity, okay? And so the main thing I want to talk about today is going to involve the fact that the genetic code, your DNA, what's called the human genome, is so complex. It is more complicated, more highly designed than anything that we have come up with so far with our human technology. All right, and think about what we are attacking. We are specifically attacking Darwinism or gradualism, because that is the controlling paradigm in the scientific community today, even though it is not very well substantiated, and even though there are very credible arguments against it. So think about neo-Darwinism. What is the gist of neo-Darwinism? What is the gist of it? It's mutation, selection. Mutation, selection. Mutation, selection. All right? And so I gave you 3 steps for gradualism. The first one is that we have variation by random mutations. The randomness is what basically William Dembski and Stephen Meyer were attacking with the specified complexity argument. What is William Dembski's field of work? What is it? It's mathematics. So specified complexity deals with the impossibility of gradualism, the impossibility of random mutations being responsible for all the genetic information that we have, being responsible essentially for the origin of life. So William Dembski in specified complexity attacks the randomness, okay, and the unintelligent, undirected processes that supposedly are responsible for the origin of life. What does Michael Behe's argument of irreducible complexity attack specifically? Accumulation of numerous successive slight modifications. All right, now today we're going to talk about genetic entropy, and we're going to be specifically attacking mutations. What did we not attack? Yes?
Natural selection.
Natural selection. Why not? We see it. Natural selection happens. And today we're going to be talking about mutations. Do mutations happen? Absolutely, and at a very high rate, a higher rate than geneticists want to admit. And to me, the idea of genetic entropy is a direct testament to the fall of man in Genesis chapter 3. Okay? What's the wages of sin? Death. Okay? And what does Romans 5:12 say about it? For as by one man, what? Sin entered into the world, and death by sin. And so death passed upon all men, for that all have sinned. And today we're going to talk about genetic entropy. It's going to be the crux of our argument today. And genetic entropy is evidence that what Romans 5:12 says did happen and is happening. It's happening right now. Okay? How did sin bring about death? The answer is through mutations. biologically speaking. Okay, so I do want to review Stephen Meyer, and I want to kind of review some of the things that you should have recognized in the Mysteries of Life DVD. What did Stephen Meyer apply specified complexity to? Information. Remember? Stephen Meyer was talking about information, and we talked about the fact that when letters convey a message, in other words, they contain information, they had to be the result of intelligence. Complexity implies design, doesn't it? Right? But information requires intelligence, and that is one of the main points that Stephen Meyer talked, how he applied specified complexity as he made the design inference with respect to information. When you have random letters, they don't convey a message. They don't contain information. So we're going to be talking today about where all your genetic information is stored, and that of course is in the DNA molecule. And we're talking about how highly complex it is. Okay, there's some, also William Dembski is the one who gave us the design inference. What happens if we apply the design filter to DNA? If we run DNA through the design filter, are we going to get what's called contingency? Do you remember the design filter? The first thing we ask ourselves is this a contingency or is it a necessity? Is it something that's just going to happen due to the laws of nature, due to just chemical attraction, or is it something that requires outside intelligent, outside forces, etc.? If it is a contingency, then it's not a necessity. Then we go to the next step. The next step, of course, is complexity. How do we detect complexity? What are we looking for if we're going to conclude that it's complex? We're looking for a highly improbable event, or a highly improbable, in the case of information, a highly improbable sequence. So then if it is a highly improbable sequence, in the case of DNA or in the case of random letters, then we look for specificity. What is specificity? It's correspondence to the English alphabet and to English words. We call that a recognizable pattern. Okay, so for specified complexity then, what are the two conditions for us to infer design? We have to find a highly improbable event. That's complexity, and then we will look for a recognizable pattern. So, specificity is the kind of the linchpin at the bottom of the design filter. We can see things that are complex but not designed. We can see things that are not contingency. In other words, they're the result of natural processes. Remember, snowflakes are complex, but they're not specified. There are things in nature that are the result of necessity, that are complex. But most of the time, that complexity is just a repeating pattern. So, we want to now center in on DNA. DNA, deoxyribonucleic acid, is the genetic information carrying molecule of the cell. Of course, we mentioned 1953, and James Watson and Francis Crick won the Nobel Prize for discovering the structure, the double helix structure of DNA. And so DNA is a vast source of genetic information, much more complex and contains so much more information than any of our computer information systems, any of our technology. And of course, information must come from intelligence. We can't have both complexity and specificity without intelligence. We have to have intelligence ordering the information, or it won't convey a message. So It's no different than software or no different than a book. Intelligence is required when you're going to convey a message. And so today's point is that DNA is a much more highly complex message than any human information system or any human library. All right, you need to be very familiar with the structure of DNA as a double helix, and you need to be familiar with the fact that that double helix is like a twisted ladder. And in the center of that twisted ladder, which would correspond to the rungs of the ladder, are 4 different molecules: adenine, guanine, cytosine, and thymine, conveniently just represented with the capital letters A, G, C, and T. So what we have here is a quaternary code. What do I mean by that? If you're familiar with computer code, it's a binary code. Zeros and ones, 2 characters. Our genetic code is in 4 letters: A, T, C, and G, and multiple combinations of those in groups of 3. So we will have 3, it's like having a word, 3-letter words. 3, they're called nucleotides. 3 nucleotides, adenine, guanine, cytosine, or thymine, will be grouped in triplets, similar to having 3-letter words in English. So if we unravel the DNA and look at a single rung, when DNA unravels, the rungs are kind of cut in half, they're split in half because they are complementary bases that are paired together. And then when it unravels, it will separate into 2 strands. One strand is called the sense strand, the other the antisense strand. And a protein will come through and read, just like you saw in the DVD, read the genetic code. All right? And so as the DNA is unwound and it's reading the genetic code, it's going to look like this, a sequence of A, T's, G's, and C's. In reality, each of the nucleotides are in triplets. It'll read AT, this little sequence that I made up will read ATT, that's one codon, one triplet called a codon. Then the next codon would be GCA, etc. So we're going to have a continuous sequence of those nucleotides, but when they are read, the process is called transcription. When they are read, for the DNA to actually produce a protein, they will be in codons. And this is literally a code. Each one of those codons has a specific meaning. It can be a stop codon or a start codon. Each one of those nucleotides will code for a particular amino acid. That allows DNA to tell the cell the exact sequence of which of the 20 amino acids will be put in a string to produce a particular protein. Okay, genetic diseases occur when there's a mistake in the DNA, and of course that mistake would be called a mutation, and one of the base pairs has the wrong letter. What happens if one of these codons has the wrong letter? It'll bring when the cell is making the protein, it'll bring the wrong amino acid. Say, well, what's one amino acid out of thousands and thousands? Well, it will cause the protein, if it's a critical amino acid, it'll cause the protein not to function. How many have heard of sickle cell anemia? That is a genetic disorder. Okay? And what happens with sickle cell anemia, everyone has red blood cells that essentially are floating little discs of hemoglobin. Hemoglobin is the protein that carries oxygen. So when your blood travels through your lungs, it picks up oxygen, doesn't it? How does it do that? On the hemoglobin molecule on your red blood cells. Well, a person with sickle cell anemia has a genetic mutation that misplaces one of the amino acids. So the hemoglobin cannot fold in on itself properly and it doesn't function very well. In fact, it's deformed to the point that the red blood cells are no longer nice little inner tubes, kind of looking like a donut without the hole punched all the way through it. That's what your red blood cells should look like. Well, what happens with a person that has that genetic mutation and the malformed hemoglobin? Their cell is in the shape of a sickle instead of in the shape of a donut. Okay, and so they have a malfunctioning protein. It distorts their red blood cells, and it also causes their body to be very inefficient in transporting oxygen. And I think it might be a little important that all your cells have adequate oxygen, right? So mutations can cause very small mutation can cause a very serious problem. So understand that DNA, which is a double helix, which when you unwind the ladder will give you a string of nucleotides. And that string of nucleotides is literally a code arranged in triplets of 3 letters. And each triplet either tells the cell which amino acid goes next, or it tells it to start the protein or stop the protein. And everything in your body is all about proteins. Your structural components are primarily protein. Your functioning molecules are also proteins. So here's the DNA, a schematic of the DNA molecule. Here's the double helix. This part here is kind of the sides of the ladder. That's made of a sugar phosphate. So it's called the sugar phosphate backbone. Here's the rungs of the ladder. You can see A always, or T, always pairs with A, C always pairs with G. And if we take out a single rung, half of the rung will be one base, this is a base pair. Half of the rung will be one nucleotide base. The other half on the other strand will be a complementary nucleotide. I'm going to keep saying complementary, and that means T and A are complementary. They come together to form the double helix. C, which is cytosine, is complementary to guanine. And so everything is in a code, a very sophisticated code. So what are the chances that mutations are going to generate new code? What are the chances? So the problem here is the whole basis of neo-Darwinism is mutation selection. Mutation, which is going to rearrange the genetic code or cause a problem in the genetic code, we're going to use that as the source of genetic information to code for new structures, new organisms. It doesn't make any sense. And the idea here is that the mutation selection idea sounds good on the surface. And it's accepted. It is the basis of biology in the scientific community. Okay? And the whole point is it doesn't work. So the complexity of DNA. The complexity of DNA is difficult for me to even describe to you. Previous years in this class, when I'm talking about DNA and I'm talking about genetic entropy, I used what I called the library illustration. How the DNA is like a big library. How in the library, what do we have here? Books. And we open up a book, what does each book have? Chapters, right? And then what does each chapter have? Paragraphs. What does each paragraph have? Sentences. What does each sentence have? Words. What does each word have? A letter, one of the 26 English alphabet letters. And how do we convey information in written form? Sequence of letters to form words, right? Sequence of words to form sentences. Now you're all going to be taking, and are taking right now, English, and then research and reports, where you're learning how to put together English information. There's nothing random about it. There's nothing random about constructing words and putting them together in sentences and then putting your thoughts together in a paragraph, right? And then organizing your paragraphs to have a coherent chapter. There's a lot of effort that it takes to write a book. It takes a lot of intelligence to write a book. Well, I'm no longer using that library illustration because it's actually very, very inadequate. DNA is much more complex than linear information, sequences of letters to form words, and then sequences of words to form sentences, and then sentences. It's much more complicated than that. because not only does it read that ATTG, read linear. When you're reading in a line, correct? DNA doesn't just read in a line. DNA is three-dimensional. It's highly complex. Highly complex. We're just now starting to understand that. In the 1990s, they began what is called the Human Genome Project, where they sequenced the human DNA. Can you imagine how much time and how many technicians that took? It took essentially a decade. It was finished in about 2001, and then they began phase 2. Phase 2, they started figuring out what part of the sequence actually codes for a protein. That project is of course, it's still ongoing. They're still filling in gaps. They're still trying to understand DNA. And they've been working on it for decades. Okay, in 2007, they finished what they called phase 2, the ENCODE project. And they found out that at least 90% of your DNA is actively being encoded by your cells. And the thing about DNA is, like I said, it's not like a book. It's not even like a computer program, because it's got 4 letters and it can be read. Do you picture in your mind what we saw on that DVD? You saw them coming into the cell with computer animation, didn't you? You saw the DNA molecule unwinding, and you saw it reading the DNA strand and bringing in particular amino acids. They showed that to you in animation, those molecules working together in concert to produce a particular protein. Well, that was showing you reading it one strand, one way. That strand can be read backwards. The strand can be read, start here and stop there. It's in 3 dimensions, so it can be read multiple ways. We can't do that with linear reading. We can't do that with reading off of a printed page. The printed page can be read one direction, one way, right? That's not true of DNA. DNA has multiple layers. So there are multiple messages in the same strand because it's 3 dimensions. Because what happens is we have a string of letters and it can read this way. DNA can also fold and change the message. It's 3-dimensional. No computer program can do that, and certainly you can't do that with written words. DNA is astonishingly complex. And yet evolutionists want to tell us that it is the result of millions of years of randomness, millions of years of mutations and mistakes, somehow getting it together in exactly the right order. And exactly in the right order, not just in a linear one-line manner, but in multiple manners. Okay, so that's the complexity of DNA. There are 3 billion nucleotides in your genome. There are 2 complementary strands. That's the double helix. They used to call them, and I guess they still do, I mentioned the sense strand and the anti-sense strand. But now they found out that both strands are being actively transcribed. Before we used to thought, okay, the DNA molecule is going to unwind and it's going to come in and it's going to read one strand. Well, it can read either strand, and it can read it this way or that way. So there's multiple codes, one on top of the other. That's not possible with written linear language. Multiple reading frames. Okay, if you read a book, does it have multiple reading frames? No, you can only read it in one way, left to right, down the page. 3-dimensional code rather than linear code. It's not just a single strand reading in one way, line by line. That's what I mean by it's not linear code, it's 3-dimensional. We don't just read it line by line in one direction. It's multi-directional. It's multi-layered. So look at this, multiple levels of overlapping codes. That's not possible with a book. That's not possible. So our library illustration really doesn't, there's a lot of information in this library, which is actually a very small one. But even that does not compare to your DNA. Does not even compare. Here's the human genome. The human genome, you know you have 46 chromosomes, 23 pairs. This is called a karyotype. If you were to scrape the inside of your cheek, you would attempt to get all of the chromosomes, and then you would have to organize them and identify them as, here's the first pair. These are called autosomes. These first 22 pairs, the first 22 pairs here, are autosomes because they control the characteristics of your body in general. Everything about you is coded in this DNA. And then the 23rd pair are the sex chromosomes, XX being female, XY being male, that determine your gender. This codes for those genes. And notice we have 2 pairs. We have double copies of everything. This is how the genetic traits of your parents, your 2 parents, come in and are blended together to form your unique genetic makeup. So in conclusion, DNA could not have evolved from random processes. Here is a review of the 2 requirements for specified complexity. DNA displays specified complexity because it is a highly improbable arrangement. Arrangement's not even a very appropriate word because it's so much more than just a linear sequence of those 4 letters. And it is a recognizable pattern. Maybe not recognizable to us because we don't know the genetic code and we don't know what each codon represents, though scientists do know that. It is a recognizable pattern. But then I want to now get back to the idea of creative design, and I want to leave behind intelligent design, and I want to now keep reminding you that intelligence, that required intelligence, is from the mind of our Creator, the God of the Bible. Okay? So now I'm actually going to begin the formal genetic entropy argument, even though I've already laid the foundation for it in transition from intelligent design. Okay, the genetic entropy argument. Here's a nice picture of chromosomes. Well, I guess we'll let this represent what? Some type of mutation. So today we're going to be attacking mutations. According to the Darwinian synthesis, the whole gist of evolution was mutation, selection, mutation, selection. All right, and so while we're going to specifically attack the idea that mutations do not work as a mechanism of gradualism. They do not work. Here is John C. Sanford. He is retired now, but he still is affiliated with Cornell University. He is a genetic engineer, in particular with plant biology. And he wrote a book called Genetic Entropy and the Mystery of the Genome. Look at this is the original cover. We've got a spaceship here and we have a library. And initially this was his cover, and he took a lot of criticism for it because it looked like it was some type of science fiction. So he actually did change the cover of the book. And he is one that would illustrate, or used to illustrate, with a library. But remember, we're going to now say the library illustration is inadequate. But his idea was that the human genome contains just as much information as would be necessary to build a spaceship or the space shuttle. Can you imagine the plans? I've seen the plans for several of the buildings on our campus here, and they're highly complex. There's pages and pages telling where every little electrical wire goes. The plans for this building would have to detail everything about where the plumbing is, where each electrical wire is, where each outlet is going to be. Very, very detailed and complex. And John Sanford says, well, the human genome would contain even more information than all of the specs. Wasn't it interesting? What are specs in common engineering and construction speak? They're designs, right? They're specifications. he is the one that really has detailed the idea of DNA being so complex. This is the same slide a second time. You need to understand how complex DNA is, and you need to understand the structure of DNA. You need to understand that it is a highly complex and even beyond our understanding. It's so complex, it's even beyond our understanding. We have the two complementary strands, the sense strand and the antisense strand, but now they're both being read. We have multiple reading frames. We have the three-dimensional code. We have multiple levels of overlapping code. And the reason I put this slide back here is because even John Sanford's idea that the specs for building the space shuttle, for example, are inadequate to compare DNA to. So here's John Sanford again. Here's the cover, the new cover of the same book. He got rid of the spaceship. And he is a geneticist. He is associated with and worked for years and years, decades, with the plant engineering, plant genetics, and genetic engineering at Cornell University. He has the patent on what they call the gene gun. That is the technique that they use to insert genes into the DNA of plants to alter the characteristic of plants. How many have ever heard the little phrase or the little acronym GMO? Genetically modified organisms. He is responsible for much of the technology that involves how they can genetically engineer plants. So he knows a whole lot about DNA, he knows a whole lot about genetics, and he knows a whole lot about how mutations work. And he has told us that the genome is not improving. If we were evolving, wouldn't we be seeing some improvement in our genome? What do we see in the genome? What do we see? Here's what John Sanford says. John Sanford coined the phrase genetic entropy. Now, John Sanford is not Michael Behe, and John Sanford is not William Dembski or Stephen Meyer. John Sanford is a committed Christian. When he gives his talks, he always brings in the plan of salvation, and he always ties into the fact that mutations are evidence of the fall of man. Mutations are why we die. Mutations explain the biological background to why we are dying. Mutations explain why we age. Mutations explain cancer. And mutations explain how it is that when God said the wages of sin is death, how it actually happens to us physically. It happens through mutations. So genetic entropy is the mutational degeneration of our genes. And here's the important thing I want you to learn from John Sanford. The fundamental reality of the fall is visible in our genome. There is so much genetic evidence for Genesis chapter 3, and there's also genetic evidence for the genealogies in Genesis. We're all familiar with, from Sunday school, the question, who's the oldest man in the Bible? Every one of you knows that answer, correct? Methuselah, 969 years. How is it possible that Methuselah lived 969 years and then we have gotten down to where we live about 70 years? What explains the decrease in our lifespan? Mutations. Okay? So the fundamental reality of the fall is visible in our genome. He calls it genetic entropy. So the reality is, notice the quotation marks. These are from John Sanford. The reality is mutations are killing us. Mutations cause aging. Mutations cause cancer. Ultimately, mutations cause death. So when God told Adam that when he ate of the tree of the knowledge of good and evil, he would surely die, guess what? He surely died, and we are surely dying either. Also, not either. So what is genetic entropy? Entropy comes from physical science. Oh no, I'm dredging up bad memories of high school. Entropy, you probably saw that in high school. Entropy is the universal tendency to dissipate, degenerate, decay, and to become more and more disordered. It's also an increase in randomness. Entropy, a universal tendency to dissipation, degeneration, decay, disorder, and it's an increase in randomness. Does this explain the complexity of the genetic code? It's the opposite of syntropy. Entropy and syntropy. Syntropy would be the increase in order. Syntropy would be creating new genetic information. And syntropy, of course, requires intelligence, an outside force. Left to itself, everything in the universe will decay, dissipate, degenerate, and tend to become more disorderly. Genetics. We've been talking about genetics. It's a branch of biology that studies DNA and genes and how they produce living things. So your genetic makeup is your DNA. Of course, DNA is made up of different genes. Genes are located on chromosomes. Much like a library contains information, well, the genome is the library. The genes would be possibly the books in the chromosomes. All of that is your genome. So genome we can define as the sum total of an organism's genetic makeup. So when I say human genome, I'm talking about all the genetic makeup of a single human being. Genome is the sum total of an organism's genetic makeup. So then we put the two together. As John Sanford did, we have genetic entropy. This is applying entropy to the human genome. So what is genetic entropy argument? The genetic entropy argument is the idea that mutations are part of entropy. And mutations are driving the human genome to degeneration, not evolution. Mutations are driving the human genome to extinction even, to degeneration, not evolution. So actually what's happening? The opposite of evolution. We are having a loss of specification by mutation. Mutations result in the loss of genetic information. That's the key. Mutations do not increase information as required by evolution. Mutations decrease genetic information. That's critical. Mutations do not increase genetic information. What do you think has a bigger genome? A bacterium or a human being? No comparison, correct? A bacterium is a single cell. We are made up of 100 trillion different cells. Not different cells, a total of 100 trillion cells. There's not that many different kinds. So, the source of genetic entropy is mutations. Mutations. Mutation is a mistake in the replication of the genetic code. And guess what? John Sanford had told us that the fact is the human genome is deteriorating. The human genome is deteriorating. How is it deteriorating? Through mutation. So mutation isn't giving us any new organisms or any new genetic information for natural selection to select. According to John Sanford, we have a continuous erosion of our genetic information. That's genetic entropy, a continuous erosion of information. So if we want to think about mutations in information, and mutations are typographical errors in that genetic code. So it's like having typographical errors in an instruction manual. Or, if we want to use a computer analogy, a mutation is like a word processing error in a computer code. The 0 and 1s are out of order in the digital code that makes up computers. All right, what are mutations? There's the definition of mutation. It's a permanent alteration in the DNA sequence that makes up a gene. Yes, it's like a misspelling. It's like a typographical error. It's like a mistake in the digital code if I'm talking about a computer program. But in reality, what is a mutation? A mutation is a permanent alteration in the DNA sequence that makes up a gene. That's what a mutation is. We're only using the idea of typographical error, misspelling, as an analogy. That's not what a mutation is. A mutation is a mistake in the DNA sequence. And a few months ago, when we talked about the evidence for evolution, we talked about the types of mutations. Mutations are permanent alteration of the DNA sequence. Something has happened to change the sequence. A point mutation, a deletion of a particular nucleotide, an A, T, A, or G, or an insertion, or a duplication, or sometimes they get moved around to different locations called a translocation. There's all kinds of possible mistakes. All of these are mutations, which are alterations of the genetic code, alterations of that sequence. Point mutations, recognize that slide? You've seen it before, except we changed the background. That's a single base pair. Remember the base pair? An A with a T or a C with a G? That's a point mutation. It can cause a change in the resulting protein. Or we can have a deletion insertion, which causes a frame shift that alters the reading frame. Oh, and remember, DNA has multiple reading frames. So we insert or delete a base pair, we're going to shift the message, and it becomes gibberish. Duplication. You can see sometimes we'll have duplication of just one little part, or we can have multiple repeats inserted. These are all mistakes, typographical errors in the genetic code. We call them mutations. All right, so genetic entropy is going to result in the loss of information. Let me show you here real quick. We'll come back to this on the last lecture. There it is. Let me just illustrate the mutation for you. This is a little part of instructions. But our DNA is a whole lot more complex than this. I copied this out of a little instruction book to put together a piece of furniture. It says fasten the center back, G, to the top, E. Have you ever tried to put something together and follow one of these? Anyways, this conveys a message. It gives specific instructions. And what happens if you put it together out of order? You're going to have to take it apart and start over. But what happens, according to John Sanford, we get 3 mistakes per cell division. Okay, so what happens? 1 cell division, 2 cell divisions. Uh-oh, I'm already going to have the wrong piece. Another cell division, another cell division, another cell division, another cell division, another cell division. What's happening to my message before your very eyes? What's happening to the message? It becomes garbled. Fast-led center part Q there's a word. Buck 17H the head cress Wagton the Swenter Buck G to the Rop Cassinga loft up it's garbled. So genetic entropy is a fact, and it best fits the creation paradigm. All right, so what we're going to do on Monday is we will have a brief quiz dealing with the complexity of DNA and a little bit about John Sanford. I will not ask you any questions from the intelligent design argument. We're going to leave that, and I'm going to come back to this and go through the slides that we didn't finish. I got a little hung up reviewing. And so we'll go through this genetic entropy argument again, and then we'll tie it up with an example of creative design. All right, we'll see you on Monday.